So plant growth regulators, like these are the factors, right? That these are the chemicals, intercellular factors, like the characteristics. Now the plant growth regulators are small simple molecules of diverse chemical composition. They could be indole compounds, that is indole 3 acetic acid IAA, adenine derivatives, that is N6-phosphorolamine, purine, that is kinetin, and derivatives of carotenoids, that is abscisic acid ABA. Now terpenes gibberellic acid GA3, and gases ethylene C2H4. Now plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones in literature. So plant growth regulators, these are the substances, these can be the plant hormones, these can be the phytohormones, etc. Now PGRs can be broadly divided into two groups based on their functions in living plant body. One group of PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern function formation, trophic growth, flowering, fruiting, and seed formation. So these are the plant growth promoters, like they promote, okay, they promote fruiting, flowering, trophic growth, pattern formation, and all that. Now which are them? These are auxins, gibberellins, and cytokinins. Now PGRs of other group play an important role in plant responses to wounds and traces of biotic and abiotic origin. Okay, they also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR, that is the ethylene, could feed either of the groups, but it is largely an inhibitor of the growth activities. But is largely, so PGR ethylene, it is largely an inhibitor. It is largely an inhibitor of growth activities. So it is not counted as promoting, but it is also growth promoting PGR. Okay, now the discovery of the plant growth regulators, you see interestingly, the discovery of each of the five major groups of the PGRs have been accidental. Now all this started with the observation of Charles Darwin and his son Francis Darwin, when they observed that the colubtails of canary grass responded to unilateral illumination by growing towards the light source, that is phototropism. I repeat it again. When Charles Darwin and his son Francis Darwin, when they observed that the colubtails of canary grass responded to unilateral illumination by growing towards the light source, that is phototropism. After a series of experiments, it was concluded that the tip of the colubtail was the site of transmittable influence that causes the bending of the entire colubtail. Okay, after a series of experiments, it was concluded that the tip of the colubtail was the site of transmittable influence that causes the bending of the entire colubtail. Now auxin was isolated by FW Wendt from the tips of the colubtails of wheat seedling. And colubtails of canary grass. Charles Darwin and Francis Darwin, don't get confused in here. Okay, now experiment used to demonstrate that tip of the colubtail is the source of auxin. Arrows indicate direction of light, so tip of the colubtail is the source of the auxin, right? As you can see, there is a site of transmittable substances, so it gets bent. Now when you cut it down, it show no bending, but when you cap it, it show no bending. But when you eliminate the light over it, so it shows that bending, right? So BCD, ABCD, right? Okay, now the Bakani, that is fully seedling disease of rice seedling, was caused by a fungal pathogen, that is Gibberella fujikurae or E. kurosawa, that is 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrates of the fungus. The active substances was later identified as gibberellic acid. Now the Bakani, that is fully seedling disease. Now Bakani, it is a fully seedling disease. Bakani disease of rice seedlings was caused by fungal pathogen, that is Gibberella fujikurae E. kurosawa. 1920s report the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrates of the fungus. So when they treated with the sterile filtrates of the fungus, the active substances were identified as gibberellic acid, so it causes the rice seedling diseases, right? Now the F. Scoop and his co-worker observed that that from the internodal segments of tobacco stem, the callus, that is the mass of undifferentiated cells, proliferated only if in addition to oxygen the nutrient medium was supplemented with one of the following extracts of vascular tissues: yeast extract, coconut, or DNA. Okay, so in addition to oxygen, the nutrients medium was supplemented with one of the following, that is extracts of vascular tissues, yeast extract, coconut milk, or DNA. Miller et al., that is in 1955, later identified and crystallized the cytokinesis promoting active substance that they term kinetin. Okay. Now, during mid 1960s, three independent researchers reported the purification and chemical characterization of three different kinds of inhibitors, that is inhibitor B, abscission 2, and dormin. Later, all the three were proved to be chemically identical, so it was named abscisic acid, that is ABA. Now, H.H. Cousin in 1910 confirmed the release of a volatile substance from ripened oranges that hasten the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene gaseous PGR. So this is it. That is H. Cousins confirmed the release of volatile substance from ripened oranges that hasten the ripening of stored unripened bananas. Right. So this is all it. Right. Then next is the oxine, then next is the ethylene, then next is the abscisic acid, then that's it. So I think we are going to cover like the oxine.